Hello everyone, Namaskar. Today in this 30th lecture of the course, we will discuss about various quality and safety aspects of juices and concentrates. Particularly, we will discuss about the evaluation of quality attributes. The quality attributes physical, chemical, rheological, and microbiological quality, how they are determined in various fruits and vegetables, juices and concentrate, we will discuss in the next half an hour or so. So let's see first what do you mean by quality. Quality comprises both subjective and objective characteristics and can be termed as fitness for consumption. A good quality material is good for consumption. It is fit for consumption. So the term quality has been defined as the totality of features relative to the ability of a produce to fulfill its requirements as determined by the producer, manufacturer, or consumers. So whatever the quality standards are set by either regulators or industry or producer or manufacturer, that is the product should meet those standards. So that is what. And how do we know that the product meets those standards? We go to analyze it either in laboratory or in some other way, that the standard protocols are there. So along with the quality, another very important attribute is safety. A food may be good in quality, but it may be unsafe for consumption. So the safety is a very, very important prerequisite of quality of any food. Okay, so what does safety means? That it's absence of any harmful substance in the food at the time of its consumption. When it is being consumed, it should not contain any harmful or any toxic component, maybe whether it is microbiological, chemical, or any other aspect, any other toxic agents. So the evaluation, now we'll talk about evaluation of food quality. Basically, it is a quality system. As I told you that these are the various attributes. Earlier classes also we discussed. So those attributes make the quality system, and the quality assurance and quality control are the two important that aspect of this quality system. Quality system is a bigger umbrella, and within this are the quality assurance and quality control. So the quality is evaluated in two ways. One is the subjective method of evaluation, other is the objective or instrumental method of the evaluation. In the subjective method, like sensory characteristics like taste, aroma, flavor, even sometime size, etc., color, all those things we evaluate subjectively by sensory, using our sensory attributes. Okay, and for this there again are the standard protocols. That is the trained panel of judges are selected. That is those judges, panelists, they are first trained about the product which is to be evaluated. That is for example, suppose tomato paste or tomato juice color is or its taste is. So the panelist who is evaluating it, he or she must know what actually a good color of the tomato is, what a good color of tomato concentrate, tomato paste is, or a flavor. So they are the consumers, panelists are, they are trained, and there are some sensory record, data record sheets are prepared. This using the standard protocol, the judges are given the sample and they are asked to record their impression about the sample, various attributes on the data record sheet, and then those data are later are analyzed using various methods. So that is how, that may be 9-point hedonic scale rating or other such statistical methods are used to analyze the data. Sometimes logic evaluation is also used to analyze the sensory data and to find out the goodness of a sample. So that is in the instrumental method or on the objective method, like pH, water holding capacity, vitamins and mineral content, and such other attributes are determined. So basically that is the in the next slides, we will discuss about what are the various physical, chemical, rheological, and microbiological quality attributes of juices and concentrates and their determination methods. So, in the physical quality, when we talk about the juices, mostly the TSS, pH, color, turbidity become the important attribute among the others. In the chemical components, chemical characteristic, it is the vitamin C because the fruits are they are good sources of vitamin C. So that is considered to be one major component. Vitamin C, ascorbic acid, a titratable acidity, anthocyanin content, a total phenolics content, like which also gives the idea about uh, antioxidant capacity. That is what is that antioxidant value of this particular fruit or vegetable juice or concentrate. In the rheological characteristics, normally STD, shear analysis is done, how the viscosity and other rheological attributes are determined. And these rheological attributes basically they help in designing the food processing equipments, etc. Then finally, in the safety attribute which we are talking about, that the microbiological quality. Microbiological quality, that is whether what is aerobic mesophilic counts, total plate count, coliform count, yeast and mold count. These are the some standard method which every laboratory and in the depending upon the product one has to use. Otherwise, one can go. There are also methods for determining a specific toxic. That is what is the toxicity value or LD50 value of microorganism, etc. And toxic contaminants and all those things can be evaluated. So let's say first we go for the physical property determination, total soluble solids. The total soluble solid value is defined as the amount of the sugar and soluble materials present in the product. Generally, fruits and vegetables contain more sugar, particularly fruits, they contain more sugar than the other solid constituent or soluble constituents, and hence the degree bricks provides the useful guide of the TSS or sugar content. The TSS is reported in degree bricks. Okay, and the measure it is measured by either refractometer, either hand refractometer or aberrefractometer. Okay, so refractometer measures the TSS concentration based on the principle of refraction of light. In a Briggs refractometer, the refractive index is calibrated into degree Briggs readings. The refractive index depends on density of solution. Measurements have to be made at a specific temperature. Particularly, normally it is done at 20 degrees Celsius temperature, or and then a suitable concentration is applied. If the temperature is high or low, then some it is multiplied by some factor, and accordingly the actual value is determined. So you can see that in the figure how that the hand refractometer is being used. First it is being sample is cleaned, and then the data is that is the standard scale in the hand refractometer. So the scale is standardized, and now the person after that it is using that is some proper glass distilled water, pure water. It is being rubbed, it is being cleaned. Okay, and after cleaning, now the sample, that is juice sample or concentrate sample, is put, a drop of that is put, and the cover is closed. After that the reading is just, it is red, that is the scale there is inside, it will give directly the degree Briggs reading. That is about how the hand refractometer works. Then now let's see the determination of pH. It is a quantitative measure of the acidity or basicity of the sample. Fruit juices have different pH levels. They can be measured by the pH meter. In the slide here, I have shown that what are the barriers, like tomato juice, it has a pH of around 4.3, apple juice, maybe it has pH of 3.3, and so on. So the pH is a quantitative measure of the acidity or basicity. Okay, the pH meter measures the change in the electrochemical potential established by the hydronium ion concentration across a semi-permeable glass membrane or an indicator electrode. You can see in this electrode how these are the electrodes. Okay, the first the equipment is calibrated, pH meter is calibrated using some base, that is buffer solution of a lower buffer, maybe in the buffer 4 or then buffer 9, and the reading is, once after the pH meter is calibrated, then the sample is taken into the beaker and then this electrode is put there. Then electrode measures the potential, that is hydrogen ion concentration, that is across, and the gradient difference in the hydrogen ion concentration, that is inside the electrode and the outside in the environment. If the difference is whether it is negative or positive, on the basis of that, that is what are the hydrogen ion gradient it is determined, and then it is represented in the form of pH, and the indicator shows accordingly that actual pH of the commodity. Okay. Next is the color measurement. The color of the fruit juice and concentrate is determined using a spectrophotometer. Okay, in the spectrophotometer, that there is a light source, and this light is that is monochromatic light, it passes through the, that is the cuvette is used, and this light, that is absorbance, what is the light absorbed, that is absorbance is red in the spectrophotometer. The colorimeter is an instrument which measures an amount of light that a sample absorbs basically. Okay, the colorimeter works by passing a light beam through a sample to measure the light intensity of a sample, and this equation one can use, that delta E is equal to L2 minus L1 to the power whole square plus A2 minus A1 square plus B2 minus B1 square whole to the power 1 by 2. And these are normally told as LAB value. You can see in the color scale, that is L, whether A value is, when it is minus, it is considered to be green. When it is plus, it is considered to be red. Then similarly, B value in the positive side is considered yellow, or negative side it is considered blue. When L is equal to 0, it is black, or when L is equal to 100, it is white. That is, so these are the color scale. Accordingly, LAB value are determined using the colorimeter, and then this delta E, that is color value, is calculated using this equation. Okay, the principle is shown here also in the schematic diagram. Okay, then turbidity measurement is another. Turbidity is the cloudiness or haziness of a fluid caused by large number of individual particles that are generally invisible to the naked eye. It is, they are similar to smoke in air. In case of juices and concentrates, this is caused by microorganisms, yeast, molds, bacteria, their presence, or even polysaccharide particles, protein, tannins, anthocyanins, or subcrystals of other foreign matters such as filter fibers, filter aids, or sand. So all these things might be present in the smaller quantity in the colloidal suspended part, or in the these particles may be. So they may give the juice, they may make the juice turbid. So the turbidity of the juices is determined using turbidity meter. Okay, you can see the principle of how it is turbidity meter works. First, the equipment is calibrated. Okay, again, that is this reading is put the sun, and the reading is brought 0. Then rotate the calibration knob to the next, to the center position. Okay, okay, adjust the 0 knob so that the, now it is showing 0. Okay, then now place the 100 NTU calibration solution, and again now is adjust the knob, rotate the knob, adjust to 100. So first it was calibrated to 0, then it is calibrated to 100. So now actual solution is put into the juice or concentrate, and it will give directly the reading it will give. Now this has the 126 NTU is the turbidity of this sample. So in this way one can find out the turbidity of a sample. Okay. Now we will discuss chemical properties. In the chemical properties, total phenolic content is very important. You know, the fruit juices and concentrate, vegetable juices, concentrate, etc. They are known for their antioxidant potential, and these antioxidant are due to the presence of various phenolic components and other. So the total phenol content of the sample is analyzed, and it gives an idea about the antioxidant capacity of the component. Phenolic compounds are important plant constituent. Okay, so the hydroxyl group in the plant extracts are responsible for facilitating the free radical scavenging. As a basis, phenolic content can be measured using Fallin coical tube reagent in the extract. So that is about 500 microliter plant crude extract is taken, and in this 300 microliter of hydrogen peroxide, maybe 1.5 molar is used, and it is properly vortexed and mixed uniformly. And after that, it is put on the FCSA. Then FCSA, you can see that to each of the micro plate, it will add about 1.5 ml of the plant crude extract treated with H2O2, or 240 ml of water, 15 microliter of 0.25 N FC agent, wait for 3 minutes, and then add 30 microliter of 1 N Na2CO3. Incubate it for 2 hours in the drink, and then read the absorbance using spectrophotometer at 760 nano. And these absorbance readings are then matched with the standard curve, and one finds out the total phenolic contents. Okay. Then total flavonoid content accordingly. Flavonoid help regulate cellular activity and find out free radicals using causing oxidative stress on the body. It helps in body functions more efficiently while protecting it against everyday toxins and sensors. Flavonoids are also powerful antioxidant agents. Examples of flavonoids are naringin, quercetin, okay, cameferol, hesperidine, neohesperidine, and poncerin present in the different types of fruit juices, etc. And so these total flavonoid content for their determination method is given briefly. It is described here in the slide. You can take 200 milligram of sample, extract it under the flux with 40 ml of ethanol, 60% volume by volume, and filter it, that you get the whatever that extract is there. Again it is a reflux with 40 ml of ethanol, 60% volume for 10 minutes, and both are mixed. You get the complete volume 100 ml, and then this is tested, test solution and compensation solution. Finally the spectrophotometer reading is observed at 401 nanometer after 30 minutes. Okay, using the either test solution method or compensation solution method. That is, the process is given here, you can read. Okay, so this is observance the spectrophotometer reading recorded, and for all the spectrophotometric readers, one has to also prepare a standard curve, and the data obtained the sample is read with the or compared with the standard curve. Then ascorbic acid determination. Ascorbic acid is a reducing agent. If a solution of iodine is added to a solution containing ascorbic acid, then it will reduce the iodine, which is brown in solution, to colorless, or that colorless iodide ion. You can see here the equation, that is iodine, which is brown. After reduction it becomes 2I, that is, which is a colorless. So in this process the ascorbic acid is oxidized, and this reaction can be used to estimate concentration of ascorbic acid in a sample. So normally you take around 10 ml of the juice sample or concentrate sample, which can be concentrate, can be thoroughly diluted. Then add 15 ml of the distilled water, take 5 ml of 1 m hydrochloric acid, 2 ml starch, and titrate against iodine solution. So this is the starch iodine reaction, and you get change in color is gives the end point, and then one can calculate how much the ascorbic acid is there. Alternatively, the 2,6-dichlorophenol indophenol visual titration method is also used for the estimation of the vitamin C content. In fact, the more frequently used method in the chemical laboratory in the food industry, it is the 2,6-dichlorophenol indophenol. So this is also the principle of this is also that this dye is reduced by the ascorbic acid, and once first one has to calculate the dye factor, that is a dye solution of standard known concentration is titrated with the known concentration of ascorbic acid, and the dye factor is calculated, and then in the unknown sample it is evaluated using the titrated using the dye, and then one can find out calculate the vitamin C content or ascorbic acid content in the sample. So in this slide I have given this is the figure that is the formula of ascorbic acid and normal vitamin C content in the common fruit juices like orange, lychee, guava, pineapple, or there is mixed fruit juices. It may range from around 15.38 milligram to about 38 or 40 milligram, like lychee juice contains around 38.46 milligram, whereas the pineapple juice may contain 30 milligram or so. The ascorbic acid is a very important component. It if it is deficient in our diet, it causes various problem like scurvy, the major disease caused by the deficiency of ascorbic acid. Its symptom may be fatigue, mood change, weight loss, joint and muscle ache, bruising, dental conditions may be bad, or dry hair and skin infections, etc. These might be a lot of problems. Similarly, sometimes if you take excessively more amount of ascorbic acid, that may also sometimes cause some problem like redness or the flushing on face, or that formation of stones in kidney, indigestion and heat burn, etc. and so on. Okay. Then the titratable acidity is another important quality attributes. Okay. It is a measure of the amount of acid or acids present in the food sample. It should not be confused with pH, which is the measure of the concentration of hydrogen ion. pH is the measure of concentration of hydrogen ion, whereas the acidity gives an idea about the amount of acid present. It is an important parameter in determining fruit maturity and sour taste in the citrus foods. Juices with a high titratable acidity are difficult for saliva to neutralize and are thus potentially particularly damaging to the teeth. So pure fruit juices have high titratable acidity, and immature fruit juices will normally have a low sugar to acid ratio. So these are and all the fruit juices, that is this is just a standard that is the acidity is found out by normal titration with the standard sodium hydroxide solution, and the acid value is calculated, that is based on the a particular acid, like in the case of apple pear it is the standard acid is used as a malic acid. In citrus fruit it is citric acid. Okay, in this strawberry also citric acid. In the grape it is the tartaric acid. So their equivalent weight of these acids is used to calculate that the titer burette reading to convert it into the percent acidity, and the acidity of the fruits and vegetable juices or concentrates are also always expressed in terms of a particular acid. That is, as I told you, in the grapes it is percent tartaric acid, in the apple percent malic acid, in tomato percent citric acid, and so on. Then anthocyanin content, they are anthocyanins are member of flavonoid groups of phytochemicals, which is a group predominant of fruit juices, concentrates, and vegetables, and this can be found in berries like blackcurrant, elderberries, blueberries, strawberries, etc. And their juices, as well as in the red wine. Anthocyanins are an important component. So these anthocyanins are rich in antioxidants and are thought to be anti-inflammatory and help boost the immune system. Okay, in the table given subtotal cyanin, total polyphenol, then antioxidant activity, and what is the how TATP ratio influences the antioxidant activity in the commodity. Okay, so for the determination of antioxidant activity, okay, there is a it is quantified generally in terms of total phenolic content like TPC, or that flavonoid or radical scavenger activity, that is RSA, or 1,1-diphenyl to picrahydryl, that is DPPHSA value, and the even total flavonoid content are also determined. And the method is given here, either DPPHSA or FRPSA is used, and in the case of DPPHSA, using the standard protocol, one can go and read the absorbance at around 5.7 nm, and in the FRAP method, FRAP assay, absorbance is read at 700 mm, and the absorbance value, then using the standard curve, is the total antioxidant capacity is known. And here in the some of the common juices like blueberry, mixed vegetable juice, etc., there is the percent inhibition of the DPPHSA, or the TEAC value, that is Torolox equivalent absorption capacity, or ORC, oxygen radical absorbance capacity, ORAC value is shown of the different juices, which gives that a fairly good idea of the antioxidant potential. Now, the rheological properties, we'll discuss that rheology is the science of deformation of flow behavior of a fluid, and particularly in the concentrate and juices, it is an important property. Viscosity is the main parameter of the fruit juice in the rheology. This is affected by the produce and system parameters. The rheology helps in the designing of equipment, juice acceptance by the consumers, and flow properties. Okay, so the normally product parameters like clarity, soluble solid content, and extraction methods are used. Our system parameters, shear rate, and temperature, and all these influence viscosity. And this viscosity data helps in equipment design. And as I told you, is the on the basis of this, one can find out whether acceptability, sometimes viscosity of a material gives an important that is the consumer's acceptance, etc., or flow property, it is indicated. So the different measuring configuration for viscosity of fruit juices is shown here. That is for the juices and concentrate, starting from the low viscosity going towards very high viscosity, either coaxial cylinder or coaxial cylinder with a recessed bottom, double gap geometry, that is this device, or pin rotors or vein rotors. They can be used to measure in the return, that is the system to measure this viscosity. And then curves, that is the viscosity versus shear rate curve, can be drawn. You can see here in this figure that different solutions, that the juices are concentrate, which have that either the dilatant or pseudoplastic, Newtonian. That is Newtonian is just, you see the parallel with the axis there. Okay, or it will be plastic or pseudoplastic with yield stress to various behaviors. And the data of the scometer, one can use the various models are there, like power law model, Wilkinson model, Cisco model, Cario, and so on, the various models. And these data can be used, and regression modeling, then again graph can be generated, and this gives an rheological where various values can be calculated accordingly. So this is here the effect of total soluble solids and temperature on the rheological properties of the juices and concentrate is shown in this figure. Okay, the increase in the temperature, as you can see in the figure, it will causes the decrease in the viscosity of the fruit juices, and this relationship generally follows Arrhenius concept which is written as mu is equal to a0 e to the power r exponential ea by rt, where mu is the viscosity at a particular shear rate, a0 is a pre-exponential factor, ea is the activation energy, and r is the universal gas constant, T is the absolute temperature. So the increase in the total soluble solid increases the viscosity of the fruit juice, and therefore if the viscosity is more or total soluble salt content is more, the fruit becomes more dense. So the concentrates normally they have higher viscosity. Then you come to the microbiological properties. The type of microorganism in juice is generally influenced by the type of microorganisms in the respective fruit and vegetable from which juice was extracted. And this there may be, if the microbiological quantity is not good, that is it may be fruit, maybe if the juice is prepared from the inferior quality of the fruits, then extraction of the juice from the bad quality of the fruit, or it is utilization of discarded fruit after harvest. Okay, if for the juice extraction non-processed water is used to make up the juice, or application of pre-contaminated water for the juice, then also it may give that microbiological higher microbial count. So these are, there is the bad quality of water used, maybe another factor. Then mechanical damage in the fruits also, sometimes that is the damaged fruits have higher chances of spoilage, or it compromises the quality of the end product, etc. Even market diseases like blemishes and decay that develop during the transit, storage, and marketing, like soft rot, beetle injury, etc. They also influence the microbiological properties, are more microbial count, and then they may lead to the toxic metabolite generation. So for the determination of the microbial number in the laboratory, in the food analysis laboratory, normally that is aerobic mesophilic count, which we also say that total plate count, that is just spread plate methods, or plate count agar media is used. So then total coliform count is also used by multiple tube fermentation techniques, or a plate count method also is used for coliform count determination, but the media is a special in this. Then yeast and mold count, here also is spread plate method using sourdough, dextrose, agar. So for all these things, that is spread plate methods can be used, but using a specific media, and even the same method can be used, similar technique can be used for getting measuring a specific microorganisms, also pathogens, etc., using standard media and standard incubation conditions. So a serial dilution method is normally used, where that sample is taken and it is diluted serially, so that you can get a statistically countable number in the plate. So the plate in this using 10 to the power 4, 10 to the power 5, 10 to the power 6 dilution is used, that is the serial dilution is used. And then this accordingly, that 1 ml of these various diluted samples are used in the taken and the petri plate. In the petri plate, specific media is used, and then media that has some saldified agent like agar agar or the things, and then it is this, once the sample is prepared in the laminar air flow state, that sample is poured and media is poured, and then it is once it is solidified, it is put into the incubator at an incubator which is set at particular temperature that is required for the microorganism, like whether coliform count or total count or aerobic mesophilic count, etc. For aerophilic mesophilic count, normally it is 35-37 degree Celsius is there. For coliform, maybe that at 28 to 30 degree. For yeast and mold count, also same 25-28 degree Celsius. Okay, and for the required duration. Okay, and after that, so basically we are taking a sample with unknown amount of these microorganisms. Okay, and then it is given with the microbial media for the microorganism, to the food for the microorganism, put into the plate, and the plate is kept in the incubator, means where in proper environmental condition is given for the growth of the. So whatever that numbers are there in the solution sample, they each organism grows there as a individual colony, and then these colonies are read by colony counter. They are counted, and then the data is expressed in number of colony farming unit per 100 ml or per 100 gram, as the case may be. So this gives, that is the normally, that the color values etc., that it should not be every commodity there are given a specific number, okay, for this amount, that yes, they should not have more than this much out, and the regulatory agencies, they have they specify normally, like in the last class we have told you that there should be nil coliform count. If the coliform count is found in any sample, it indicates that the sanitary conditions are not properly maintained in the production plant or processing line and so on. So this gives now the, I will summarize that as quality parameters are very, very important aspects, and this should be standard laboratory methods and protocols should be used to analyze the various quality attributes, both physical, chemical, microbiological, and by then many times either as a processing aid or these values are written on the label are to meet the requirement of the regulatory agencies to have the more sale value. This quality analysis is done, and quality this data of these various attributes are used as per the requirement. So these are the references which are used in this lecture. This, thank you very much for your